Okay, hello everyone. Thank you so much for the invitation. I'm going to talk about experimental molecular assembly estimations for space exploration using gas chromatography mass spectrometry. And I would like to start off by acknowledging my collaborators, Dr. Gabriela Rees, Angela Chang, and Dr. Heather Graham. There was a very exciting press release in January 2025 where NASA announced that they found the ingredients of life in fragments of a lost world. And this was a press release that showed what organic molecules they found in the asteroid return mission from the asteroid Bennu. And they truly had some amazing findings. They found some precursor molecules such as ammonia and formaldehyde in water. They found salt and clay minerals, amino acids, 15 of the 20 proteogenic amino acids, and also nucleobases. And for this mission, the goal was to learn more about life as we know it from Earth. But when I first read this title of this press release, I really would have liked to add a little side note. NASA finds ingredients of life as we know it. And when we think about life as we know it, we need to be really clear that we're actually talking about a sample size of one. We only have one example. And this is kind of the reason why this is one of the reasons why we're interested in life detection and why we're interested in life detection of other planets, because we want to learn more about other living systems. And for me personally, I would find it way more interesting to find actually a living system that looks differently to us. And in order to find a living system that looks different to the one life example that we know, we need to be able to actually detect the ingredients of life, not just as we know it, but also agnostically with as little earthly bias that we can apply. So this is kind of the reason that I'm interested in agnostic biosignatures and want to find a way to recognize life, even if it looks very different to the life we know. And because we're going to talk so much about biosignatures today, I thought I'm going to just throw up a quick biosignature definition from the NASA Astrobiology page. And this page says that a biosignature is any characteristic, element, molecule, substance, or feature that can be used as evidence for past or present life. And it needs to be something that can't be made without the presence of life. So now that we're all agreeing on a biosignature, we can go forward. And biosignatures are kind of a little bit of a rising topic right now. There was a paper from 2023 by Malater where they looked into how much the term biosignature was used in the literature between 1970 to 2021. 1970, obviously, this was the Viking era. And there's a little bit of linguistics there where back in this area, the term biosignature was not used as much. They more used life detection. But we can see now from 2010, kind of 2015, that the term biosignature was increasingly used. And I would actually say now for 5 years later than 2021, it probably is still increasing. And so with this amount of research into biosignatures, we also have a lot of papers, and when we look into these, we might get the idea that a biosignature could be pretty much anything. And so I ran through a lot of papers and compiled a random list of objects and molecules that were proposed as biosignature methods. You're very welcome to pick your favorite here, but all these biosignature methods that are proposed also lead to some biosignature claims. One of the famous biosignature claims from the past, obviously, is the 1976 gas evolution experiment from the Viking mission. Then we had the Allen Hills meteorite that had evidence of primitive Martian life from 1996. There was methane on Mars in 2004, arsenic life in 2010, phosphine on Venus in 2020, methane on Enceladus in 2024, and we had DMS on exoplanets in 2025. And we can also see that the cadence of these biosignature claims has increased. So I think as a community, we also need to make sure and think about how we make sure that the biosignature claims that we communicate are also robust. And there was a standard of evidence workshop where the workshop report was published in 2022, where they proposed 5 questions to make your biosignature claim more robust. And I really like these questions, so I'm going to show them to you here. The first one is, have you detected an authentic signal? Is your signal of good enough quality for what you would like to record? Then, have you adequately identified the signal? So this is definitely a question. Have you measured what you think you've measured? And I remember there was some controversy, for example, on the phosphine on Venus claim, where some research said that it was actually not phosphine. So the third question is, are there abiotic sources for your detection? And this goes back to the question about what's the abiotic background for the planetary body you're studying. It sounds like a small question, but this actually entails a lot, and it probably will require quite a lot of research and knowledge of the planetary body that you're investigating. And then the fourth step is, based on kind of that knowledge, is it likely that life would produce this expression in this environment? Is your finding logically sound? And then the fifth step would be, are there independent lines of evidence to support your findings? And this could, for example, be laboratory experiments on Earth that you could use to further have evidence for your findings. And because I'm talking so much about one single biosignature method in this seminar, I want to make one point very clear. I believe that no biosignature method is going to be ever standalone. We will need an array of complementary lines of evidence for a robust biosignature claim. And when I talk about one biosignature method, I always envision it to be part of a bigger array of different methods. So with the basically out of the way, let's talk about agnostic anomaly detection. And this idea is actually not new. It goes back to 1965, where Lovelock proposed that life can change the distribution and assembly of objects on a planetary body. And these anomalous changes in the composition and concentration of these molecules in the environment could be used as a biosignature tool. And so Lovelock showed this here with hydrocarbons, where he plotted hydrocarbons and their abundance in the order of their carbon number. And here in purple, you can see the abiotically produced hydrocarbons, where we see that the abiotic produced hydrocarbons are actually usually more smaller, and they are produced kind of in the same length range, where the biological hydrocarbons, we have actually much larger hydrocarbons that are produced, and the spread of different hydrocarbon sizes is also much, much wider, and we see certain hydrocarbons are produced over others, and this really shows some kind of evolutionary selection mechanism here, and this kind of behavior is what we want to be able to measure with agnostic biosignature methods. So when I think about a biosignature method I want to work with, I already mentioned I want it to be agnostic. I also want it to be testable. I don't want to propose a theoretical measurement. I really want to show if it works in the lab. And I want it to be employable in the laboratory for origin of life experiments on Earth and on other planetary bodies in our solar system. And the argument that it needs to be employable for origin of life experiments is kind of very personal requirement, because this is kind of my scientific background. I started out in origin of life, and I think a robust biosignature method should be able to quantify the success of origin of life experiments on Earth, as well as quantify the complexity of samples on other planetary bodies. And A nice to have for a biosignature method would also be easy computation, because if we ever want to have it on a rover, we will have very limited computational power. And so a method that ticks a lot of these boxes is assembly theory, which is a theory that was first proposed by Lee Cronin and Sarah Walker. And it's based on the idea that only living systems can produce objects over a complexity threshold in abundance. And here the abundance argument is really important, because this entails that we assume a biological system is producing these objects or storing these objects, and this is why they're in abundance. So this is kind of capturing this biological selection argument here. Then derived from this is the term molecular assembly, where the objects we're talking about are molecules, and we can pretty much use it as a complexity scale for molecules. And because molecules is what I measure with an instrument in the laboratory, I'm going to talk about molecular assembly going forward in this talk. There is a theoretical route for molecular assembly calculations, which is based on the number of joining operations required to build a molecule. So this is purely a mathematical calculation. This is not based on any synthetic pathway. We're starting out with the single bond of a molecule, so with our MA1 step, and then we add bonds and unique steps as much until we reach the full molecule. We're only counting unique joining operations, and if we have joining operations that repeat each other, then the joining operation only is counted once. If we do this for the molecule chrysin, here in the example, we would have 7 steps, and so the molecular assembly number for chrysin is 7. As I mentioned, I'm only going to talk about the experimental molecular assembly work, but there's been a lot of work on the theoretical part of assembly theory. And so if you're interested more in this part, I can highly recommend to look into any of these papers. There's been so much work been done in the past few years. So there's lots that you can look up, be my guest, but I'm gonna focus on the experimental part going forward. So there is an experimental route for molecular assembly estimations using mass spectrometry. This was also proposed from the group in Glasgow, and it's based on the number of fragments that you see during analysis. So here in our example, we have the ATP molecule on the left, and the more bonds and parts we add to this molecule, the more fragments we see in the spectra on the right. And because we're quite a diverse audience, if you know what a mass spec is, you can lean back for a second. In case you've never heard of a mass spec, I'm going to give a small introduction because I'm going to talk a lot about this. So mass spectrometer, if we use one, the first step is quite often chromatography. Here, as an example, I use a gas chromatograph, and if you see the plot at A, we can split up our sample across time periods so that we have over time the different analytes of our sample split up. So this would be what the chromatography step is doing. We also could just skip this step, and then we would use direct injection for our sample. So these are basically 2 options. We use chromatography or do a direct injection to put our sample into the mass spec. Then the first step in the mass spectrometer is that we need to ionize our sample, because what we measure is ions. And today I'm going to talk about 2 different ionization mechanisms. One is called electrospray ionization, which is considered a soft ionization method, which allows that the molecular ion is going to stay intact, so we can measure the full intact molecule in our first full scan. The second ionization method I'm going to talk about today is electron impact ionization, which is called a hard ionization technique, because we're separating our sample from our ions from the sample with a charge. And so we might actually not have an intact molecular ion, even if we run the first full scan. This is kind of the biggest difference. But then once we ionize our sample, we can guide it through our mass spectrometer. The quadrupole is basically a big filter, so we can filter our ions until we kind of concentrate them in the C-trap and put them into the orbitrap, which is the detector on this kind of mass spectrometer that I'm showing. There's several different detector types that we can use, but In this talk, I'm mostly focusing on Orbitrap, and an Orbitrap is a particularly sensitive detector, which is also quite suitable for untargeted workflows. And so then we would get our molecular ion. An Orbitrap, in fact, is so sensitive that we can also measure isotopes, but this is completely irrelevant for this talk. So a mass spectrometer measures the mass-to-charge ratio M/Z of a molecule and their fragments, and every molecule has a very specific fragmentation pattern, which we can then use for the characterization of that specific molecule. And we can use qualitative and quantitative analysis for mass spectrometry, while qualitative is slightly more common, and we need our molecule to be ionized. I mentioned chromatography earlier. If you run a chromatography step, for example, if you run a chromatogram of an alkane mixture, you would get your total ion chromatogram, which might look a little bit like this. So we get all our different molecules split up over time, and then we can pick kind of one of the molecules, one of the peaks, and then we would get the spectrum of this peak. And this would be the first full scan spectrum for the specific non-decaying peak. A possibility with Orbitrap that we can also do is to run tandem mass spec. So there we would, after we run one full scan, select our molecular ion. For non-decaying, we would use the one that's the main ion, which is about 268 M/Z, and then we can fragment it again and would get another spectra of this particular ion. And so this is a possibility with Orbitrap. And the group in Glasgow did exactly that. They have a route for electrospray ionization mass spectrometry for molecular assembly estimations, where they run a full mass spec scan, and then they pick their top X mass spectral peaks. I think in the first paper they used the top 10 peaks, and then they run the tandem mass spec on these mass spectral peaks, and they get their MS2 spectra. So this would be the second fragmentation step, and then they count these peaks in this MS2 spectra, and this would be the molecular assembly number referring to that specific molecular ion for that molecule. They've run this method on a range of different samples. They had some prebiotic samples, they had some E. coli and seawater, a surprising amount of whiskey and beer, some yeast samples, some inorganic samples, also some Murchison extract, and some sediments. And they were really able to distinguish the different sample types that they measured, and they also saw kind of a transition spot between abiotic and biological samples that lay somewhat around 15. The value 15 is definitely not instrument transferable as far as we know. More work would need to be required to actually know where like an overall transition value would be for, but for this sample set, for this instrument, we know it's just over 15 basically. There is more work done on the molecular assembly experimental side from the Glasgow lab, where they also measured NMR and IR and also compared it with the direct injection electrospray mass spectrometry. But the majority of these measurements were done on pure molecules, but we can see that there's actually quite nice correlation. However, with direct injection, there is a little bit of a problem, because if we imagine we want to measure samples that are quite complex on an unknown environment, we might want to have some chromatographic separation first to just improve what we see. A chromatographic step that they tried in Glasgow was liquid chromatography, which is not deployable in space in planetary settings because you would need quite a lot of solvent. However, there is actually quite a lot of interesting work that's been done. So far, it's only out as a preprint, but they used molecular assembly with liquid chromatography, electrospray ionization, mass spectrometry, and were able to distinguish phylogenetic species using this method. So on the plot on the left, you can see the molecular assembly-derived phylogenetic tree, and on the right, you can see the consensual genome-based model, and they look pretty similar. They were also able to distinguish different domains with this method. If you're interested more in this, I can recommend to check out the preprint. I think it's really nice work. But as I mentioned, ESI liquid chromatography will not be able to fly in space. So we need to use a mass spectrometry type that we can send into space for molecular assembly estimations. And luckily there is actually gas chromatography mass spectrometry, which is a spaceflight ready measurement technique. Gas chromatography separates our molecules by the interaction with the stationary phase and the temperature gradient, and it's the only technique that provides the required level of species separation for unknown environments. And it has spaceflight heritage. It's already deployed with the SEM instrument on the Curiosity rover on Mars. It's also planned with the MoMA instrument on the Rosalind Franklin rover, and it's planned to be used on drums on Dragonfly on Titan. There is a difference though when we compare direct injection on liquid chromatography mass spectrometry with gas chromatography mass spectrometry. And the biggest difference is the way we ionize our sample. So I already mentioned electrospray ionization, which is kind of the big block box here in black. And we can measure a wide range of different molecules with this method. We can measure molecules that are quite high in weight, and we can also measure some non-polar ones, a lot of polar molecules, and a lot of very polar molecules. This is very different with electron impact ionization, where the molecular weight that we can measure is actually pretty small, comparable small, and the molecules are also rather non-polar, and polar molecules are already a little bit tricky. So this is a limitation, and we had to work around with this, and we came up with a method that kind of allows us a workaround, where similar to the Glasgow method, first we run a full mass spec scan with our method. And then, because we have this limitation that because electron impact ionization is a hard ionization technique, and we might not have all our molecular ions in our full scan, we integrate our chromatographic peaks first as kind of a way to separate our single molecules from our complex sample. And then we integrate that time period basically and count the mass spectral peaks of the average spectra of that retention time window and take this as our molecular assembly number. And why this is just adapting a method for a different instrument, there are a lot of consideration that we had to make on the way. We had to compile a sample library that kind of reflected what we actually wanted to measure. Column selection was also a big part because when we use a GC column, we basically add a filter to our method. And columns are usually made for targeted analysis where certain GC columns are made to see certain product groups. So they're not necessarily done for a targetless workflow. So we might even consider using an array of different columns. Instrument settings is also an important one. It can really change what values you see. For our sample library, we used extraction methods and solvents. In a spaceflight setting, this would not be possible. Then I already mentioned the ionization method. The sample matrix can also impact what you see. The choice of instrument certainly makes an impact on your analysis. And then there is also the topic of derivation, which I'm going to talk about in a minute. On the data analytical choices, the peak detection and baseline filtering definitely had an impact on your estimation. So this really needs to be well established. We found that thresholding was a really important point here, and we really played around quite a while to find a threshold that cut enough noise away, but allowed us to still preserve enough sample information. And blank subtraction is something that we did not find useful for our clean laboratory experiments, but if we consider a spaceflight scenario, this might be actually required. And so we came up with this electron impact ionization molecular assembly method then, where we have our total ion chromatogram, integrate the specific peak, and then we have our spectra for that retention time window. We count the peaks, and this is the molecular assembly number for this method. So now that we have our pipeline, let's talk about data. Excuse me. So the first data point that I want to show you is of the polyaromatic hydrocarbon naphthalene, which we tested on different instruments, columns, and mixtures. When we calculate the theoretical value for naphthalene, we get a number of 5. So then we went ahead and tested naphthalene on a nonpolar column on our Orbitrap instrument as a single compound standard. We got a count of 56. You might now be confused that this value is so much higher than the theoretical value, but this is kind of normal for electron impact ionization mass spectrometry. The theoretical value is just a mathematical calculation. We would actually not fragment our molecule to the last possible bond. We also might get some other fragments on the measurement. And it's not a problem to have a higher number experimentally as long as we can calibrate for it with a calibration curve at the end. So then we went forward and measured a low polarity column again on the Orbitrap, and this time we used a mixture of different polyaromatic hydrocarbons, and we got a similar count to the single compound standard of 57. And then we tried the same non-polar column again, same instrument, and we ran a small molecular mixture. So this was an assortment of small molecules, and we get a slightly higher count here of 87, which kind of might suggest that we had some interaction in the ionization with other molecules present. And then as a comparison, how this method would perform on a completely different mass spectrometer, we used a low polarity column again and tested a TSQ. This is a mass spectrometer that's more suitable for a targeted workflow, and the mass analyzer is completely different to an Orbitrap, so we would actually expect that the result would look quite different. We measured the polyaromatic hydrocarbon mix again, and we got a count of 236. So this really shows that this method is not just instrument transferable. We would need to calibrate every specific instrument we're using. So then we went ahead and we stayed with the Orbitrap and we measured an NRK standard to kind of start off. And so in this plot, you see our molecular assembly estimation value on the left, on our y-axis. On our x-axis, you see the molecular weight of our known standard. We applied a threshold of 1E7. I mentioned this needed a little bit of trying to get a good threshold, but 1E7 was the one that seemed most suitable. Our experimental data is shown in blue and the theoretical data is in pink. And what you can see now is that it's really hard to actually see if we have any similar correlation because there's this experimental versus theoretical calculation gap. So we normalize this data and now we can actually compare the data set. We just use the standard minimum maximum normalization, and we see that the alkanes are not following exactly the same trend, but it is quite similar. With this, we went on and tested a full sample library. We tested different aliphatic, aromatic, and chlorinated compounds. And the experimental trend line that you see here in green is the trend line that is representing all 3 experimental test groups. And then we also calculated the theoretical values, which is shown again in pink. And we really show that for volatile molecules, the trends for the electron impact ionization molecular assembly estimation are quite similar. So for volatile molecules, this seems to work really great. But if we go to a different planet, we're truly not going to come across volatile molecules. So what are we doing about this? A very common method for making your non-volatile sample volatile in GSFC is derivatization, where you add functional groups to your analyte to make it more volatile. An analogy for this could be that your molecule is a little caterpillar, and in order to make it more volatile, you add wings and make it a butterfly so that it flies. And so a common derivatization method could be DMS, where we add little cilia rings to our molecule. TPDMS is also used by the SEM instrument on Curiosity, but there's a little bit of a drawback with derivatization because the number of added functional groups can vary depending on the molecule properties, and it's not completely fully controlled. And this might actually not just be a problem for molecular assembly estimations, but a general problem for untarget diagnostic analysis, because we are taking a sample that is very unknown in the beginning, and then we're kind of modifying it in a fairly untraceable manner. So this might be a problem. To investigate this further, I took some theoretical calculations for the molecular assembly values of the 20 protogenic amino acids, and I plotted them in the order of their non-derived form. So this is the blue box. And then I took 3 common derivatives, TFA, TMS, and TBDMS, and also calculated them for the correlating amino acids. And as we can see here, unfortunately, the derivatives are not really following the linear trajectory of the non-derived molecules. And this would also suggest that if we look at these amino acids in a non-targeted way, that we might not necessarily recognize what we're actually looking at. I also done TBDMS derivation in the laboratory of some prebiotic plausible molecules and some amino acids, and this is truly the worst plot I'm showing you today. There was absolutely no correlation, and I really tried, but I'm pretty sure that molecular assembly estimations on derived samples are really not possible. But there is a workaround with this. There are GC columns that we could potentially use for slightly more polar compounds. We, for example, in the lab tested a stubble box column where we were able to measure Krebs cycle intermediates and fatty acids. And this also leads us to the question, when we do molecular assembly estimations, we are not aiming to understand every single molecule in our sample. We really truly want to understand the complexity of the sample in an untargeted way. So might it be possible to just quantify the complexity of a sample while only looking at the non-polar compounds? And to test this, I looked at an experiment that the group in Glasgow did. They cultivated yeast for a couple of days and then burned it at 200, 400, and 600 degrees and measured it with electrospray ionization direct injection Orbitrap. And they saw a very clear transition between the living and degraded yeast. This was one of my absolutely favorite experiments that they did in this paper. It's in the SI, which, yeah, this is my favorite plot in the whole paper. I really like this, and I wanted to test this with our method and if we could actually see the same trend. And indeed, we actually saw a very similar trend. We also saw that our native yeast was the highest data point we measured and was degrading with the increased baking times. We also measured our samples in triplicates, and we see quite high error bars, and this is really representing, we went back into the sample and saw the different specters. It's representing quite a heterogeneous sample mix of already broken building blocks and polymerization products in the 200 and 400 degree sample. But when we come from a biosignature side again, this is also a cautionary tale, because when we want to recognize a biosignature, we don't just want to find a life, but we also want to find potentially altered remains of deceased life or degraded life. And so this experiment also shows that this might be a problem with this technique. But we went forward and measured a few more complex samples to really test how our electron impact ionization molecular assembly estimation performs. We did an extract of the meteorite Murchison to show meteoritic organic molecules. We did an extract of spirulina to represent prokaryotic organism. We did an extract of yeast to represent an eukaryotic organism, and we took a GCMS gasoline standard of which represents fossilized organic molecules. And so when we look at the Murchison sample, this kind of shows the lowest molecular assembly estimation count, which we would expect because it's kind of representing the abiotic background. The prokaryotic organism is the sample that is showing the highest count. It's interesting that it's quite different to the eukaryotic organism. This might be something with what we can extract from the cells, but if anyone has an idea why they're so different, I find it really interesting and would love to hear some theories. And in the gasoline standard, you might be quite shocked that it's almost as high as the eukaryotic organism, but this is a clear measurement bias because this is a GCMS standard. It's specifically made to do gas chromatography measurements, so all the molecules in the standard are readily available for this measurement. If we've used a different kind of sample done an extract, it potentially might be a little bit lower, but here the sample is so high because there's definitely measurement bias here, but we kind of have a little bit of a curve of liveliness in our different sample types. And with this, I'm actually going to slowly wrap up. I find it really important to also include limitations when we think about a new method. And I already mentioned this, but choices such as instrument, columns, methods, and data processing have a considerable influence on our final molecular assembly estimation. So care really has to be taken with this. And when we calibrate for an instrument, we really need to establish a stable set of these settings. We have the limitation with this method that we can only measure volatile molecules that are ionizable by electron impact ionization. So far, we've seen that this seems to be not a massive limitation, but it's definitely there. And I also really want to reiterate that no live detection method is standalone. Molecular assembly can reveal environmental anomalies, but we will not get a definitive live detection answer, and we would need to have an array of complementary methods for a definitive proof. And to wrap this up, electron impact ionization molecular assembly can be used to detect anomalies in the molecular assemblages in an unknown environment. A little bit as a kind of outlook idea, developing agnostic biosignature methods for already deployed instruments might offer a new opportunity in biosignature development and the interpretation of already collected data. And now if you ask me, well, what's the point? Why is this supposed to be useful? The benefit I see in this molecular assembly estimation is that we're able to take a very complex sample set, such as a mass spectrum, and we can transfer it into a number. And actually with a fairly simple method. So we need fairly low computational power to transfer our spectrum into a number that then a computer can read and make decisions based on this. And this might be specifically interesting with the current push towards AI-driven automated data evaluation on other planets and AI-driven rovers, for example. When we think about rovers that are quite far away, where communication to Earth might not be super without any delay, we might want to be able to already make decisions on the fly of potential measurements that we additionally take. And so if the molecular assembly value could be high, this could be leading to more measurements, or it could actually also help to decide what kind of data we want to be downlinking to Earth. If a spectrum has a particularly high molecular assembly value, this might be a good target to be sent down to Earth for then humans to look at and then decide how the mission would be going forward. This is kind of how I imagine this could be particularly useful in space exploration. But with this, I'm thanking all of you for listening, and I'm very happy to take questions.
So my question is regarding the potential discovery or detection of macromolecular aggregates of organic matter. So for example, the Cassini, when it flew through the Enceladus plumes, detected all of these large high molecular weight organics that are unidentified and are possibly abiotic. And also, I don't know much about this, but possibly the Titan dunes that are made up of organics might have a lot of complex macromolecular aggregates of organic matter that formed abiotically. So my question would be directed to that. How could molecular assembly theory cope with this kind of abiotic large organic matter that could possibly yield very high MA values?
Based on what I know, I think I don't quite know what the concentration of these large molecules are, but this might already be filtered out by the argument that we're looking for higher complex molecules in abundance. So this is kind of why the abundance argument is so important, because we're kind of saying that an environment would select to build these specific molecules. And intuitively and based on what I've read so far, I think these larger molecules would not be made in abundance for that particular environment. So this is kind of how we would get around this.
All right. Yeah, I don't know the abundance. I don't remember relative to the actual other detections on the cosmic dust analyzer. They were abundant, but this was of course relative to other detections. So in that same instrument. So compared to other measurements and other environments, I don't really know. But on Titan, I also don't know. They might be super abundant because
But
it's full
again,
of organics.
when we look for Titan and they're just part of the abiotic background, then they would not be the anomaly for that environment.
Of course. Yeah, that's true. Well, thanks a lot.
You're welcome. Thank you.